Welcome to our lecture on protein. So we've seen this page before. We've talked about carbohydrates and fats already. So in this lecture, we'll go through proteins as the third of our macronutrients. Again, we know that fats, proteins, and carbohydrates are similar to each other in that they're these large molecules made up of smaller molecules, which we call monomers. We know that all three of these macronutrients contain the elements carbon, hydrogen, and oxygen, and therefore they carry energy in the form of calories. We'll see on the slide that proteins differ quite a bit from, especially carbohydrates, but a bit from fats as well. So carbohydrates, you'll recall, primarily serve as a source of energy to the human body. Fats can be a source of energy. They're the secondary source of energy. But then fats are also really critical for, again, hormone function, transportation within the blood, digestive capacities, and the structure of the cell membrane. So we'll see with proteins have a lot of very important functions outside of being a source of calories. So when we look, if we were to look at like aspects of the human body or even aspects of plant matter under a microscope, we'd see that so much of what's going on is happening via proteins. So the structure of plant matter, the structure of human cells, just like with fats, it's made up of proteins. We see, and once we look inside of a cell, we'll look at those various organelles inside of a cell. The structure of those is made up of proteins. One of the main functions of protein in the human body is acting as enzymes. And these are basically little chemical molecules that encourage reactions to happen. And so later on in subsequent lectures, we'll look at how enzymes in the body affect metabolism, our digestion of our food, and then how we utilize the byproducts of digestion, how the body manages fluid balance, how we transport nutrients through the blood. Similar to fats, some hormones in our bodies are made of proteins. Immune cells are made of proteins. And then this last line here is just a difference, kind of calling out another difference from fats and carbohydrates, is that proteins, in addition to carbon, hydrogen, and oxygen, also contain nitrogen. And we'll see that some amino acids contain sulfur. So here are building blocks of proteins. We call them amino acids. You'll see the structure again having that carbon-carbon sort of backbone, but again, pretty significant structural difference compared to the carbohydrate and compared to the fat. So two major parts that differ are this addition of the amino group in the amino acid. So this is the nitrogen-containing component of the amino acid, and then the side chain, or we call it the R chain, and this again will be any a chain again of carbon molecules with some oxygen and hydrogen, and then in some cases the side chain will also contain sulfur. So this is a little bit similar to our the structure of our basic carbohydrate molecule, where we bind glucose molecules to each other to form more complex carbohydrates. So a similar analogy here, we take amino acids and bind them together to form peptide chains. And we can actually talk about roughly talk about the length of the chain by calling it a dipeptide, 2 amino acids, tripeptide, 3 amino acids, oligo, and polypeptide, where we start to have, hundreds of amino acids bound to each other. So really when we're talking about protein function, we see long polypeptide chains bound together. And so here we'll go through, proteins don't really exist just as peptide chains, but the peptide chain is sort of the fundamental component of the protein. So when we talk about the total structure of the protein, we actually we kind of have to go through 4 steps. So we're talking about the primary structure of protein, the secondary, the tertiary, and the quaternary structure. So in the primary structure, we have just the peptide chain. In the secondary protein structure, we have that peptide chain, it starts to twist or fold on itself. And then tertiary structure, the peptide chain starts to do even more twisting and folding on top of its twists or folds, kind of taking on now a very three-dimensional shape. And in the quaternary structure, we see two or more polypeptide chains binding together, and that's actually finally where we have a molecule that we call a protein. Here, I think I have two slides of schematics of this. So again, the primary structure, it's just your amino acid polypeptide chain. The secondary structure, when I said bending or folding before, that's literally technically called beta pleated sheets is one way to make that bend or twist, and then the alpha helix is the second way to make that fold or twist. And then in the tertiary structure, you'll see you still have the beta pleated sheets or the alpha helices, but now with farther on itself. And then, sorry, I jumped to the next slide unintentionally, but if I go back, it erases what I recorded. So we'll just stay here. But again, we'll focus in this slide on the quaternary structure then. So you'll see, and I actually do like this slide better to showing the quaternary structure anyhow, because it's, you see that now we have 4 different polypeptides bound together in the quaternary structure. You can see that color-wise. So that's most of what I want you to take away. That's all of what I want you to take away for the structure of proteins. And so again, I guess to sum it up, just understanding that the polypeptide chain in the primary structure is really just the beginning of making the full protein. And really, we don't have a functional protein until we get to the quaternary structure. And you'll see, we won't talk about it so much in this lecture, but because this structure is so complex and intense, it does require pretty specific conditions to be functioning. So as soon as we start to change the environment too much or the conditions, we can see loss of function in proteins. So now we'll go through a little bit talking about how proteins are made. So how do we even get to that crazy quaternary structure? And again, with this content, I have a few slides to follow. So all of the proteins that we have in our body that we make are already coded for in our DNA, in our genes. And so when we basically then read the genetic code that's written in our genes and read that code and figure out what proteins to make. So each different tissue matter in the body will read different parts of the genetic code to then produce the proteins that that tissue or that part of the body needs. And so we call this reading of the genetic code gene expression. So gene expression is the production of proteins based on the genetic code. We know that you might recall from basic biology that our DNA is made up of molecules called nucleotides. And these nucleotides are written into sequences in the DNA, and specific sequences of nucleotides call out for specific amino acids. So when we're talking about reading the DNA or genetic expression, it's kind of like reading a book, how we have different letters form different words. And so we know what that word is based on what letters are in the word. It's kind of the same thing. The sequence of amino acids indicates the sequence of the nucleotides, kind of like the letters, indicates a particular amino acid, kind of like a word. So that's how we figure out gene expression. And I'll show you the images on the next slide. And then the last piece of protein synthesis. So first we have gene expression, just knowing that the sequence for the amino acids is coded for in the DNA. And then we have transcription and translation, and this is where we actually copy the sequence of code from the DNA into what's called messenger RNA. And then from the messenger RNA, we start to build the amino acid chain. Here is a little bit of a blurry schematic, but not horrible. So you see we have the nucleus on the left-hand side, and inside the nucleus we have the DNA. So if we were to unwind the double helix of DNA, we would actually see the nucleotide expression here. So, and then you'll actually see on the left side the codon. So the codon is basically the word. So we have GCG. That is the codon for one specific amino acid, which we'll see next down here. So what's happening on this left side is that the messenger RNA is basically making a copy of the nucleotide sequence. And I know this doesn't look exactly like a copy, but there are like alternating pairs of nucleotides, G and C alternate and A and T alternate. And so what's going to happen is actually once we get to translation, we're going to see this flip again. So it's kind of like mirroring. But anyway, we transcribe the nucleotide sequence of the DNA, creating mRNA. And then we leave, the mRNA actually leaves the nucleus, you see, and goes out into the cytoplasm of the cell and goes to one of our organelles that's called a ribosome. And it is at the ribosome that we see the process of translation. So here you'll see in the ribosome the mRNA sequence, again with the codon. And what you'll start to notice now is that each little 3 nucleotide sequence is going to be matched by a tRNA, a transfer RNA, which carries an amino acid with it. So here's where you see the mirroring back of the nucleotide sequence. And so basically it's kind of as simple as that. So we start to build this amino acid chain based on the nucleotide sequences that we're reading now from the mRNA. And you'll see the newly formed protein chain. You start to have these are different amino acids that we see down here bound together into a polypeptide chain. And then up here you kind of see, this is like the more dramatic schematic of the proteins, the full polypeptide chain coming out, and then even seeing a little bit of the bending and folding. So that's the basics of transcription and translation by which we make proteins. So I haven't talked too much yet, or really at all, about the amino acid pool. So when we look at that last slide on translation of the mRNA sequence, then to make the polypeptide chain, those amino acids have to come from somewhere. So we know that we store some glucose as glycogen in our muscle and our liver, and we can store fat in our adipose tissue. We don't really store protein too much, kind of obviously because it's doing something functional. So we don't just kind of hang on to it for later, but we do continually break down old cells and build new cells. So what that means is then we have some amino acids sort of always at the ready to use depending on what we need. We call that the amino acid pool. It's not a super large supply, but it's just there is constant turnover of tissue matter, which means there's constant degradation of enzymes and hormones and proteins. And when we degrade those, we harness the individual amino acids and then can use them to make different proteins or make new proteins. You'll see some of the things that play into this amino acid pool: tissue protein, plasma protein, enzymes, hormones, antibodies, hemoglobin. Again, so your blood, your white blood cells, hormones, enzymes, and then tissue matter can all be broken down, and the amino acids go into the amino acid pool. Dietary protein, so definitely when we consume foods that contain protein, most food contains protein, those amino acids go into the amino acid pool. And then non-protein compounds, heme and heterocyclic amines. We don't need to know that so much for our purposes of our class, but knowing that this pool is sort of continually added to. And then you'll notice a couple of these arrows that only go away. So actually all the arrows except for dietary protein also move away from the amino acid pool. So in the same way that we might break down tissues, enzymes, hormones that contribute to the amino acid pool, we'll pull from the amino acid pool to build new tissue and proteins and hormones and enzymes. Similarly, we will use from the amino acid pool to metabolize or break down food, or again, break down tissues, break down hormones, break down enzymes. Water, urea, and energy all require some amino acid protein contribution. And then we can use some amino acids to actually, not the amino acid themselves, but component of the amino acids to make glucose or even to make triglycerides if we have excess. Really only if we have excess would we be doing this. Here's just another schematic of that, which I think this is a little bit more simplified. So you'll see again, body proteins and dietary proteins contribute to the amino acid pool. From the pool, we might see protein synthesis or synthesis of nitrogen compounds. So this is actually a really important piece of proteins and amino acids, is that this nitrogen, remember the amino group of the amino acid, we don't have much need for this, and so we do actually have to get rid of it. So we'll convert the amino group into ammonia. That's usually happening in the liver. And then ammonia is actually a fairly toxic compound, so we do need to get rid of this. So we'll convert this ammonia to urea, and then we'll pee urea out. So we have liver and kidney acting to remove the nitrogen compounds when we have excess amino acids. And then we can take amino acids, and we're going to look at this word in a subsequent slide, but we can actually take some amino acids and transaminate them or convert them from one amino acid to a different amino acid. And then certain amino acids, which we call keto acids, can be used for energy, and again, could be converted to glucose, a component of them, or could be converted to triglyceride, really only in excess, when we have excess amino acid. And the only way we have excess amino acid is from overconsumption of dietary protein, or if we were really sick and breaking down our body proteins. That's really the only way to get excess. Okay, so here's where we actually see the different names of the amino acids. So we have, depending on the source that you look at, we have roughly 9 essential amino acids, and then we have about 11 non-essential amino acids. So roughly 20 amino acids. Again, remember, nutrition is an ever-evolving science. So some of the latest research will say we've discovered about 24, 26 amino acids. But for now, these are the 20 that we know pretty well. So essential amino acid, as the diagram shows, these are amino acids that the human body cannot make. Therefore, we do have to eat these. We have to consume food, and the food will provide us with these essential amino acids. And then the non-essential amino acids, our body can make these through that process of transamination. So we can either take a non-essential amino acid and make another non-essential amino acid, or in some cases take an essential amino acid and make a non-essential amino acid. So here is that process of transamination. You don't need to know the chemical conversion, not for this class, but just know what transamination is. It's taking one amino acid, alanine, and converting it to another amino acid, in this case glutamate. And again, this is why some amino acids are not essential to be consumed in the diet, because we can make them. So essential and non-essential only is in relation to the diet, the foods that we eat. All of the amino acids are essential for life, but the essential amino acids it is essential that we consume them in the foods we eat. So then just kind of reviewing this slide again, but not much to say here, just remembering the reasons for why we have protein in the body, and they're plentiful reasons. And then the last two, I think I just have two more slides here just to kind of give you some more diagrams and imaging on our amino acids. So you'll notice, again, all having at least a 2-carbon backbone, and then the amino group, and then some sort of R chain, which may or may not be very long. So you'll see that some of them, the R chain is an actual like hexagonal ring or a double ring. And you'll notice that some of these, cysteine and methionine, also contain that sulfur molecule. And then again, you'll notice some of this terminology we don't need to know for this class, but you'll notice again the amino acids from that previous slide with the full ring around them, those were the, I believe, non-essential, and the broken ring were the essential. But refer back to that slide as you like. It's a really, I think, a really nice diagram. And then this one is just I found this just online, but the data is good. The information is good. Looking at our 8 essential amino acids, so leucine, isoleucine, and valine, lysine, methionine, phenylalanine, threonine, and tryptophan. This is a neat little selection of the foods where we would find these essential amino acids, and then what role these amino acids play in the human body. So I'll let you look at that on your own time. And that's the end of this lecture.